what we in my lab are interested in is understanding what kind of molecular mechanisms shape chromatin fibers in three-dimensional space. And even though mitosis is something that I studied in grad school and postdoc, and they always recommend you to switch subjects for your independent group, I still kept working on the subject of mitosis, dedicating a small fraction of lab's time to this question, specifically because mitosis is a fantastic lab for understanding the mechanisms that act on chromatin fibers, because it represents this state of extreme compaction. To my knowledge, to the best of my knowledge, there is no in vivo state that would be more compacted than mitotic chromosome. Well, actually, there is one, that's sperm. Because in this state, the cell has to do everything that it's in its power to shrink chromosomes so that they're easily segregatable from one another, and so that it could send them into two daughter cells without any kind of error. So that presents a nice opportunity. And actually, arguably thanks to mitosis, we understood three key mechanisms that, or forces that act on chromatin fibers in 3D on a large scale. So it's in mitosis that we discovered sister chromatid cohesion, namely the linkage between sister chromatids via SMC complexes cohesins. Arguably, it's mitosis that initiated the line of thinking about loop extrusion. For example, I would argue that it's John Markov who really introduced, who drove the last iteration of introducing loop extrusion to the field. And he, in his seminal work from 2012, he and his postdoc, they tried to argue that loop extrusion is necessary to explain formation of mitotic, compactization of mitotic chromosomes. And then mitosis is among the subjects that also suggested that chromatin must be able of undergoing phase separation. And there's been a lot of research on the subject over the last 5 years, including the one presented in this conference. But so one would argue that, well, we have a lot, why keep studying? But I think few things remain completely unknown. That is, first, how these mechanisms interact with one another, namely how cohesion, loop extrusion, how loop extrusion phase separation interact with one another to give the shapes of mitotic chromosomes as we see them under different methods of visualization. And the second question is whether that's all. Whether that's it, we have three mechanisms to ever talk about, or there is something else that's missing, and we would need it to explain the transformation of chromatin. Like many of you, I come from a physics background, and there is a very well-known mantra that what I cannot create, I do not understand. And I think it's actually a really nice tool in evaluating the state of knowledge. Namely, if you can reproduce something, it argues that maybe you have enough. It's not, it doesn't guarantee that, but it argues for that strongly. And like many of you, I do that in silico. So in the remaining of my talk, I will try to reproduce a mitotic chromosome from these forces in silico to answer these two questions, whether we understand interactions between different mechanisms and whether we know everything. And what makes mitosis complicated is that when it comes to SMC complexes that drive 2 out of 3 of these mechanisms, mitosis has 4 of them. Mitosis starts by condensins 2 getting onto chromosomes, then condensins 1, confusingly, condensins 1 come onto chromosomes later. As shown in this diagram, it's initiated by, prophase is initiated by condensin 2, and when the nuclear envelope breaks down, condensins 1 come in. But also there are cohesins, of which there are confusingly 2 types as well. There are cohesive cohesins, that is cohesins that cohese chromosomes, keep them together. And there are cohesins that potentially keep extruding loops. And when it comes to cohesive cohesins, we know that they're very well present there in prophase, but gradually deplete throughout mitosis. And when it comes to extrusive cohesins, it remains somewhat unknown. It's clear that they should be there at the beginning of prophase, but what happens to them throughout mitosis is not very well known. So the question that we wanted to answer within this study is whether we understand the structure of chromosomes formed by individual condensins, if we could isolate, just have a system where one type of condensin acts on chromosomes, and whether we could understand that, and whether we understand fully the interactions between these four types of SMC complexes. And the problem with mitotic chromosomes is that they're very difficult to study. They have complex, irregular structure that is dynamic. It develops rapidly on the time scale of minutes. And it has many molecular players that come together to form the structure. So that's why, on one side, mitotic chromosomes historically have been one of the most original subjects of molecular biology. On another, we keep talking about these chromosomes and unknown factors about them 140 years later, after the initial work. But it also argues that in order to understand them, you really need collaborative studies where you combine different methods, different lines of thinking, together and try to crack this problem from multiple angles. And that's why now 9 years ago, we formed an extremely productive, extremely well-oiled collaboration, initially between Decker, Mirny, and Initial Lab, and later as I formed my own lab, I balanced the transatlantic distribution of weights and joined this collaboration is now an independent lab. And together, because we join our forces, we can use very different methods. So from the Initial Lab, we have excellent methods for optical microscopy and electron microscopy and mass spec now. From Decker Lab, we have, in Job Decker and his lab mates, we have arguably one of the most leading experts on high C in the world. And we, together with Lenient, contribute to it with our theoretical thinking. What also enabled this study is the system for synchronization that was developed in Irnshaw Lab, where they found a way to form tightly synchronized populations of cells that undergo mitosis and that can be synchronized at different time points. And the tightness of this system enabled extremely high resolution. We could take time points as frequently as 2.5 minutes and see massive differences between them. And second, again, thanks to the enormous library of mutants that they produced in this lab, we can selectively turn off different players, in this case SMC proteins, and we could study the action of individual SMC players as well as interactions between a chosen pair of SMC complexes. Wait a second. Oh, I opened a wrong presentation. Good catch, Anton. And I realized that because in this version, because I have a few minutes more than before, I can put their faces on. So in this project, the theoretical part was contributed by my own lab and together with Leonid and Samir from MIT. The experimental part was led by, the experimental part on this side was led by Kumiko Samejima in Irnshaw Lab, and the high C was driven by Johan Gipkus in Job Decker's lab. And it's a massive study. Unfortunately, I cannot do justice in presenting all sides of this story, so I'll only focus on theory side, but soon we're gonna have a preprint where all the sides of this research will be equally highlighted. So the first question that we wanted to understand is how individual condensins fold chromosomes. Like I said, there are two types of condensins, and on top of it, we have cohesins coming in. So what we figured is worth doing is turning off 3 out of 4 of these SMC complexes. So we only have one condensin left, in this case, condensin 2, or separately, in a separate cell line, we would just have condensin 1 compacting chromosomes. Similar studies have been done before, but previously cohesins couldn't be turned off. But now we could really isolate just one type of condensins. And in agreement with previous studies, microscopy showed that when we have just condensin 2 active, chromosomes become shorter than normal, than wild type, and thicker. And thanks to the efforts of Fernando Cisneros Soberanes from Bill Earnshaw lab, we also now have an electron microscopy view of the same chromosomes. What they did is that, as much as I understand, they were able to slice chromosomes into thin layers and then reconstruct envelopes of these chromosomes by systematically going over each individual thin layer. So we have this nice view of them. We can quantify, we can measure their volumes and densities, things, and their shapes at extremely high resolution, thanks to all these enormous efforts. And similarly, looking at condensin 1 only chromosomes, as it was known before, condensin 1 forms long and thin chromosomes that are surprisingly really wavy. And in electron microscopy independently confirms the same phenotype. And in Hi-C, arguably not unexpectedly, we also see differences because Hi-C does, as we now understand well, agree with optical microscopy. Specifically, for this short and thick chromosomes, we see really broad interaction profiles, and with some features I'll describe later, while for these thin chromosomes, we see that each locus interacts just with a number of neighbors up to a relatively short window and then doesn't interact at all. Okay, so what kind of structures does it correspond to, and what can we learn about the action of condensins from that? This side, to answer this question, we turn to modeling. And we iterated over many different models, and arguably modeling is the reason that this work took us 5 years to bring together. But we actually arrived to a very relatively simple model with just 4 fundamental assumptions. First, we assume that we start with a fiber of nucleosomes. In this model, we just have a collection of particles, each representing a single nucleosome that are connected together into a freely jointed chain. And after being at this conference for 4 days, I realized that this assumption is almost insulting to half of this audience, because you folks have spent your career showing how intricate folding of nucleosome fibers is, and then here we are saying like, freely jointed fiber. But we do have to make these assumptions because of the multiple scales that the model aims to bridge, and maybe that actually represents the future directions for us. So second layer, to represent the action of condensins, we know that they extrude loops, so we immediately simulate the final stage of loop extrusion for condensins 2. That is that we fold this fiber into a collection of consecutive, randomly sized loops that are essentially back-to-back. And we know that should be the final state of condensin extrusion, because condensins 2 are extremely stable on chromosomes. So they should extrude fibers, collide with one another, and then form a stable array of loops. Then to represent the compaction, presumably compaction by phase separation, we further constrain this array of loops, of polymer loops, into a cylindrical, cylinder-like shape. And that's sweeping a lot of questions under the rug, and I'll explain what kind of questions those are and how we would like to address them in the future. But for now, that's a simplifying assumption that chromosomes are cylindrical from the outside, so we enforce them to be cylindrical. And finally, the last feature comes from Hi-C. And as I mentioned, in Hi-C for condensin 2 only chromosomes, we see these funny patterns where besides the main diagonal that represents of interactions of nearby loci, we also have 2 more diagonals. Specifically, we have these bright bands that go through Hi-C at every single chromosome at the distance of 17 and 34 megabases. Just to refresh, every diagonal in high C represents all pairs of loci that are separated by the same fixed distance that just is represented by the index of this diagonal. So these two diagonals have indices of 17 and 34 megabases. And what that could represent, we thought a lot. Oh, pardon. So one important thing about mitotic chromosomes is that they have high C that is extremely uniform. It's the flattest, most boring high C that we've seen so far. Doesn't have any kind of position-specific features. It's just this kind of diagonal decay and a banded pattern. So it turns out that these maps can be nicely summarized with a single one-dimensional curve that summarizes contact frequency, that is the intensity of interactions versus separation. And because it's high C and it has a nice dynamic range, both in terms of distances probed and contact frequencies probed, you really have to plot it in a log-log scale to fully appreciate and understand these curves. So for this specific data set, the curve looks like that. And the second and third diagonal that I mentioned look like these non-monotonic bumps in the contact frequency at 17 and 34. And several years ago, we scratched our heads a lot about what that could represent. And then it turned out that there was a feature in mitotic chromosomes that's been observed on and off in some preparations from the beginning of the 20th century, essentially, from first third of 20th century. And that feature is the formation of helices. People argued for a long time that it's because it's preparation specific, it could be an artifact, yet it's a feature that would repeatedly pop up in papers as you go back in history. So we figured that this feature could nicely explain, or at least qualitatively explain, this periodicity. Namely, if you consider loci that are separated by half a turn, their interaction frequency would be low. And then if you consider loci separated by a full term, all of a sudden, the interaction frequency should go up at least by some amount. So we, in the set of models, we forced the helical trend, but as opposed to our previous studies, this time we did it extremely softly. The way we did it is that we put an invisible rod through a chromosome and wound the array of loops around this invisible rod. And it's invisible because it doesn't exclude chromatin, but it does exclude the axis of the loop array, the backbone of the loop array. So essentially, you wind the chromosome on a spool and then make sure that it doesn't relax through topological constraints. But the resulting loop arrays are not nicely structured, as I will show you, but instead they're fairly irregular. One could argue that that's a fairly complex model. I mean, it's just for assumptions, but if you think about it, it should come with a lot of parameters. But we figured out that there is a really nice way to reduce it to a manageable, to reduce this number of parameters to a very manageable number. So first of all, we figured that experiments could be used to extract at least two parameters that are critical for this model. The first one is volume density of chromatin. And I mentioned that we have really nice, really high resolution electron microscopy images. And they show that in the case of this chromosome, we can measure volume density of the genome in these compacted cylinders. And it comes down roughly to a number of 1 nucleosome per a box of 16 by 16 by 16 nanometers. And it was roughly the same, a bit more compacted for other mutants. Then the second parameter that we could borrow from our experimental data is the length of a helical turn. Like I said, we have a helix, and because we expected already to create periodicity at high C, we can go into high C, figure out where periodicity is, and then plug it back into the model and fix it and not touch it again. That helped us to reduce the exponentially growing space of parameters, but yet we were left with 4 parameters that we couldn't constrain otherwise. So the first parameter is the average loop size, that is how big are the loops created by condensons. The second is how big are the spacings between consecutive condensons, and it proved to be an extremely important parameter that we didn't appreciate initially. That is, we learned that condensons do not come close enough as we previously thought. And then the last parameter is the helical pitch. That is the vertical step between the turns of the helix. And the technical parameter that is proved to be easier to calculate is the contact radius. The thing is that one thing that we still don't know about Hi-C is how close to loci should come in three-dimensional space for Hi-C to register it as a contact. And it's not even something that you can estimate a priori, but we figured that there is a nice computational way to incorporate the idea of a contact radius into a model. And if we sweep through it, we see a very clear effect on our simulated Hi-C data that we can use to infer it then from experimental data. So basically, we can sweep over 4 of these parameters, out of which one can be swept on CPU, and the other 3 have to. The other 3, you really need to recalculate this model over and over. Unfortunately, it's fairly expensive. We haven't figured out to make this model more manageable. So in this case, for a full sweep for the publication that we're working on, just for that space of parameters, we had to use 8,000 of GPU hours. But luckily, Biological Institute that I work at somehow provides fantastic computational opportunities, better than I have seen before in physical institutions. So the resulting set of simulations can be represented as a cube, because there are 3 parameters that we sweep, and the 4th one that we, so I guess it's a hypercube. But you can choose 2 axes to project this cube onto, and then for the other 2 axes, just mention what's the best matching parameter. And then as a result, you get those maps that basically tell you how nicely these simulations with these sets of parameters match Hi-C. And it turned out that there is a very clearly defined peak of 3 cells, this resolution of sweeping parameters. And for the best matching simulation, we managed to reproduce Hi-C across 4.5 orders of magnitudes in length and 4 orders of magnitudes in contact frequency. Namely, it means that we actually match different structural features of different sizes within the same model. And the best looking model is shown here. As I mentioned, a few things to prove to be interesting about it. Well, first of all, the best matching size of a loop agreed super nicely with mass spectrometry data that suggested that there is roughly 2 to 3 condensins 2 per 1 megabase of chromatin fiber. Then we, without plugging it into the model, we reproduce the geometry of chromosomes. Namely, we reproduce the width and linear density of chromosomes as it's seen under electron and optical microscopy. And this time we were very happy to see that the helices that we forced on these chromosomes didn't have to be perfect. In fact, it's not that easy to recognize a helix in this image. Yet, I mean, if you were to plot angular positions of each locus versus their position along the genome, there would be a clear signature of a helix. What's important about it is that optical images in vivo rarely show nice spirals. And the argument that we would make then is that we don't need a perfect spiral in the projection, but instead this helication that we're talking about is should be used is apparent only in the bulk of chromatin. And finally, as I mentioned before, we figured that gaps between condensins should be relatively considerable. It turned out that this model would only produce nice match if we assume that gaps between consecutive condensins were at least 80 nanometers big. And that's not something that we thought about initially, and that we probably wasted a year of our time forcing these gaps to be tiny, but it would never produce a match. But in the end, it actually nicely reproduced punctuated staining of condensins that was previously seen in optical microscopy. How much time do I have? So, all right, then I'm gonna skip a huge chunk of the talk. I would just mention that we figured out, using a similar approach, we figured out how condensins one-fold chromosomes. And there we use similar principles, except for with two modifications. First, because these chromosomes were so flexible and wavy, we could no longer constrain them into a cylinder because that would ignore an important aspect of their shape. And instead, we put it in the periodic boundary conditions at the fixed density, which allowed us to simulate wiggly chromosomes. And then second, we extended them slightly because we figured that in vivo, spindle would pull these chromosomes a bit and extend them beyond just a random walk style of conformation. And similarly as before, through sweeping, we also found a narrow combination of parameters that would reproduce the experiments. And again, independently, without plugging this data into the model, we figured out that this model nicely agrees with the predictions of mass spectrometry on condensing frequency, with the geometry observed under electron microscope, as well as continuous staining this time of condensins that was also seen under a microscope. And together, the thing is that in wild type, these condensins do not act independently. They come together, as shown in this diagram. First condensin 2 comes in, then condensins 1. And this time we were able to study how they work together by depleting cohesins. So we could see a structure that's unperturbed by cohesins so that we could better understand the interplay between two condensins. And there we would start with the model number 1 that I mentioned for condensins 2 and just add, without changing parameters, a layer of condensin 1 loops on top. And there is a nice explanation for that because condensins 2 come in first, they're stable, and then condensins 1 come in and would re-extrude loops that are already extruded by condensin 2. They would re-extrude them into their own loops that would be smaller in size because there are just many more condensins 1. And without changing the parameters obtained in the previous model, if we just put them together in this one and sweep just over geometry, because geometry should be able to change, we were able to reproduce experimental results equally well. So we have learned a lot about how condensins work with one another, but in the interest of time and nutrition, I'm gonna skip the second half of my talk where we looked into interactions between condensins and cohesins. Please ask me over lunch if you're interested. Yeah, sure. So, yeah, I know, right? Long story short, extremely short, we learned that condensins must be able to bypass cohesive cohesins and jump over them, while for extrusive cohesins, they should be disrupting them and kick them away from the positions occupied by them in interface. And I would argue that we have really good evidence for that from optical microscopy and high-C. I guess the last point to mention is that within this modeling line of questions is, well, what's next? To my taste, I could not see any kind of big gaps in understanding when it comes to the bulk of mitotic chromosome, but the biggest questions are posed by the devil, as was mentioned before, namely by the surface. I still do not understand what happens on the surface of these mitotic chromosomes so that they do not stick onto one another, and they make somehow force these chromosomes to be cylindrical instead of spherical. And that's something that lots of people work on as well, and I believe that we have a student in my lab, Vladimir, who may have good ideas on how to approach this question and what kind of effects could simultaneously answer both question number 1 and 3. And at the same time, there is also a question of why these chromosomes are helical in the first place. It's something we impose, not something that we derive from first principles. And maybe topology or non-equilibrium effects could play a big role there. Finally, I would like to thank the massive team that worked on this project. Like I said, it's been a lot of fun. I think the personal connections that we build over years is it's the friendships that we made along the way that value the most. And it also was the reason that I stuck to the subject. Specifically from the lab of Job Decker, it's Johan who drove the high C. From the lab of Bill Renshaw, it's Kumiko and Fernanda and Itaru, who drove electron optical and mass spectrometry. And from Mirna lab, it's Samir, who simulated lots of effects that I unfortunately had to throw away from my presentation in the interest of time. And I would also really like to thank my lab. Over the 5 years, we grew and we studied a whole variety of different subjects. And we have, like I mentioned, now a certain subgroup in my lab studies these mitotic chromosomes, but we go much beyond that. And thank you for your time. Yeah. They're allowed to intersect, but not, I mean, they're topologically transparent, but there is excluded volume. I think, so we're talking about poor solvent, right? There is, if you just look at sheer numbers, there are good reasons to believe that these chromosomes are in poor solvent. That is, they're undergoing phase transition to a liquid-like state, and whatever for polymer is liquid. Repulsion would not be enough. I mean, we've played with very simple models where you start with these bottle brushes and add a certain amount of attraction, and you see a nice collapse from them being these classical Alexander-like brushes with lots of repulsion, very stiff backbones, down to spheres in a tiny zone of interaction energy. And I don't believe that nature fine-tunes. So I don't think it's something like nature selected that interaction energy should be 0.3, 0.5. So I do suspect that there's something else, and it's not just me, right? I mean, but personally, I do suspect that there must be some other effects that prevent collapse into a sphere, and that make them cylindrical, and also prevent the collapse of adjacent chromosomes onto one another. And there are actually, there are many different hypotheses that one can already investigate. There have been ideas that maybe there's good evidence that there are soap-like molecules that cover chromosomes. These chromosomes are heterogeneous, so it's different things that we're currently investigating. Well, I don't. It actually is accidental, I think. Yeah, it's positive, but it's just because I prefer plus over minus. I think in nature, these chromosomes can be of either handedness. And there is really nice images from, I think, early days of electron microscopy, where they show two sisters side by side, and one has one helicity, handedness, and another has another handedness. So it's a spontaneous symmetry breaking. Whether it's in this field, it's called perversions. Whether when handedness changes all of a sudden, we played with it. I think IC is not terribly sensitive to perversions. If you have one per few turns, they don't really show up because the bulk of this diagonal comes from a few complete turns. So if you have like a couple complete turns, then a perversion, then back, you don't really contaminate this nice periodic signal with the signal that comes from perversion. So we're talking about tens or percents, but that's log, right? So. Yeah. Condensins too are extremely stable. To the best of our knowledge, I mean, papers that go back in 2000s, I think, show that their binding time to chromatin is similar to that of prophase. Namely, it's limited by the dynamics of this process. So if they come in all at the same time, and if they collide with one another, which is now argued in the field, but under these two assumptions, they would each bind in its own place and then extrude up to one another. And then once collided, they just would close gaps. And if you close all the gaps, you just have a series of loops of exponentially distributed length. And then nestedness you form if you have dynamics, and especially if you have different populations with different stabilities. Oh. Yeah. Yeah. So yeah, it's a nightmare question for me because I think especially after this meeting when I realized how complex this behavior could be at short length scales. And we always kind of hid away by arguing that we mostly reproduce contact frequencies at hundreds of kilobases away, where you can always take an approach of a physicist and say that, oh, it's just, if we jump over different length scales, it doesn't matter. But I don't think it's an extremely consistent position in this case because we argue that we reproduce contact frequencies in all range of separations. So I think if we were to incorporate that, these specific parameters would change for sure. To what extent we'll be able to reproduce, still have a good match, I'm not in a position to say without another 16K of GPU hours. Yeah.